Hey guys, I'm going to review an in-depth explanation of photosynthesis today. So let's jump right into it. The basic chemical equation for photosynthesis is light plus 6 carbon dioxides plus 6 waters, which yields 1 glucose, which is C6H12O6, and 6 oxygens. The main process of photosynthesis occurs in the leaves of a plant. So here's my artistic rendition of a leaf. And a leaf is made up of plant cells. So this leaf right here, like maybe this is a plant cell, and then right next to it a plant cell, and other materials as well. Much like this plant cell with its organelles and cell wall. More specifically, photosynthesis occurs in the chloroplast, which looks something like this, which is an organelle present in plant cells. The chloroplast is double membrane bound. As you can see, here's the outer membrane and here's the inner membrane. The interesting thing about a chloroplast being double membrane bound is that this is evidence for the endosymbiotic theory, which suggests that photosynthetic bacteria were acquired by endocytosis by early eukaryotic cells to form the first ever plant cells. So there's that common ancestor theory of evolution coming in right there. So coming back to photosynthesis, this is the structure of a chloroplast. Inside the chloroplast, there's stroma, so that's kind of like the liquid around these little stacks of thylakoids, so each of these discs, and then stacks of them are called grana, and this is the main place where photosynthesis takes place. The process of photosynthesis can actually be broken down into two distinct reactions. Light-dependent reactions, meaning they require light, and then light-independent reactions, meaning they don't require light, and in this case, by light, I mean sunlight. Let's cover the light-dependent reactions first. So the light-dependent reaction takes place in the thylakoid membrane inside the chloroplast. So let's say this is the chloroplast, here's a stroma, and here's this stack of thylakoids, again, like I said, called the grana, and then the light-dependent reaction takes place in the membrane of a thylakoid. So if I were to blow up this thylakoid, picture something like this, out here is stroma, and then inside is the inner thylakoid space, and this portion is the thylakoid membrane. In the membrane of the thylakoid, there are a chain of molecules that make photosynthesis happen. This chain of proteins is repeated hundreds of times around the thylakoid, although only one complete set is illustrated here. So as you can see, here's the phospholipid bilayer, as showcased by the phosphate head and then the lipid tails. And then here's the series of proteins that make up the chain of molecules that allow photosynthesis to happen. But again, just a quick reminder that this chain of molecules is repeated over and over again inside the membrane of an actual thylakoid, but for simplicity's sake, only one completed set is shown here. So, the first molecule, quote-unquote first molecule, present in the electron transport chain, which is the name for this chain of molecules, is called photosystem 2. And interestingly enough, it's called photosystem 2 because it was discovered second. So, photosystem 2 is always accompanied by the water-splitting enzyme. It's not really pictured here, but here, let me just draw a water-splitting enzyme. And the job of the water-splitting enzyme is to split 2 waters into 1 oxygen molecule, 4 hydrogen plus ions, as well as 4 electrons. When sunlight hits a leaf and the sunlight eventually makes its way to photosystem 2, as showcased here by this lightning bolt, the light acts as a pocket of energy, which excites the electrons and thus energizes them. Photosystem 2, being very electronegative, can take up these electrons as pictured here with this electron moving up the photosystem 2. The electrons are passed along the complexes, each complex more electronegative than the next, so that each complex basically oxidizes, meaning it removes the electrons from the previous complex, and current complex is reduced, meaning it accepts the electrons. And this process of reduction and oxidation and reduction continues onwards and passes along the electrons from complex to complex, until eventually the electrons make it to photosystem 1, where light once again hits photosystem 1 and excites the electrons once more and keeps them going on their journey. The main difference in the two photosystems, aside from photosystem 2 being accompanied by the water-splitting enzyme, is that photosystem 2 primarily absorbs light of the wavelength of 680, whereas photosystem 1 absorbs sunlight with the wavelength of 700. One quick thing to mention before we move on to the final reduction part of the electrons is that every time the electrons get passed through the complexes, for example, when it gets passed from PQ to CYT, a hydrogen ion is kicked inside the thylakoid membrane from outside out here. So this white space, think of that as stroma, and then every time an electron is reduced, meaning it's moved along the complexes, a hydrogen plus ion is kicked into the inner thylakoid space. This comes into play later as we want to build up a high concentration of H pluses inside the inner thylakoid space. So, coming back to the electrons that have once again been excited in photosystem 1 and have been moved up, these electrons are finally reduced by ferredoxin, that's this complex right here, and the electrons move right here. And then in a last step, they are once again reduced by this complex right here. But in actuality, this complex, it less reduces the electrons themselves and more helps NADP plus, accept the electrons, and in turn, it combines with an H plus to make NADPH. This complex right here is actually called ferredoxin NADP reductase, which again, apt name because that is exactly what this complex does, is it helps NADP plus be reduced, meaning it accepts the electrons so that the electrons have somewhere to go at the end of this chain of electrons. There's actually a lot of NADP pluses just floating around out here inside the stroma, and we'll actually get to why in just a second, why there's so many NADP pluses floating around, just waiting to accept the electrons at the end of the electron transport chain. So far, this chain of events has achieved two things. One, we've made a lot of NADPHs due to the NADP pluses accepting the electrons at the end of the electron transport chain, and consequently we've also made lots of H pluses inside the inner thylakoid membrane. Not only did we make H pluses through the water splitting enzyme, which made 4 H pluses, but we've also brought in H pluses from outside being the stroma, into the inner thylakoid space through the complexes as the electrons were being reduced, so as they were moving through the chain. So now we have a high concentration of H pluses inside the inner thylakoid space. Lots of H pluses. It's a lot more acidic. It's actually almost 100 times to 1,000 times more acidic inside the inner thylakoid space, which is all the way right here, as opposed to the stroma. Just a quick reminder, something being acidic just means it has a high concentration of H plus ions. And briefly going back to the equation that we've looked at the very beginning, with light plus 6CO2 plus 6H2O, the light part and the H2O part is dealt with in the light-dependent reaction. So as you guys can see right here, the light part comes in right here, and then the H2O part right here comes into play, and this is also where the O2 on the product side of the reaction is produced. But as you can see, the carbon dioxide has not been dealt with, nor has the glucose, but that's coming later in the light-independent reactions. So, we've established now that there's a large concentration of H pluses inside the inner thylakoid space, and you might be wondering, hey, why are we building a proton gradient to begin with? Well, as all things biochem, gradients usually don't want to be gradients. They want to go back to the way things were before, when everything was even and equal, and there is no gradient. So as such, all these H pluses that we built up inside the inner thylakoid space want to get out. But since they're polar, despite being super small, they cannot cross the phospholipid bilayer, because as we know, only small non-polar things can pass the bilayer. So the only way that these H pluses can get out is through the ATP synthase, which is like this protein right here. And the only way that H pluses can come out here and then reset kind of the gradient is if they go through the ATP synthase. Well, as they go through the ATP synthase, it kind of works like a crank, and the ATP synthase turns so that it turns ADP plus a inorganic phosphate into ATP, the energy molecule, which is what we want. So the whole point of the light-dependent reactions is to make lots of NADPHs and then lots of ATPs to be used later in the light-independent reactions. So I'm actually going to show you a quick video or a quick clip of the ATP synthase actively working, and this is a more realistic interpretation of what the ATP synthase kind of looks like. And here is also a short clip of all four complexes of the electron transport chain kind of working together. This process of making ATP using the proton gradient and also like this electron transport chain is called oxidative phosphorylation. So the oxidative part of that name comes from the fact that the electrons, again, there's that cycle of reduction and oxidation, right? And that's that part. And then the phosphorylation part comes from this, where the inorganic phosphorus is kind of added to, or phosphate is added to the ADP and turns into ATP. That's that phosphorylation part. It's where oxidative phosphorylation comes from. In the case of low NADP plus concentration, which can happen sometimes when the Calvin cycle slows down and not enough NADPH is used up, the electrons enter a cyclical flow. Because if there's not enough NADP plus, then there's no where the electrons can go, because at the end of the electron transport chain, the electrons are accepted by the NADP plus, it's reduced and turned into NADPH. But if there's no NADP pluses in the first place, then the electrons have nowhere to go. And so what happens instead is the electrons enter the cyclical flow. So they'll go to the NADP plus reductase, but then they'll go back to photosystem 2 and then travel through to the ferredoxin and then back to the reductase and then have this like cyclical behavior. Alright, so that was all the light-dependent reactions. Next up is the light-independent reactions. As you've probably noticed, there's a specific orientation of the various molecules in the membrane so that the NADPH and the ATP are expelled out into the stroma. For example, see how like this is the inner thylakoid space. The NADPH that's made is out here in the stroma. The same is the case for the ATP. The ATP is made so that it's out here in the stroma. That is because stroma is where the Calvin cycle takes place, and the Calvin cycle is the light-independent reaction. The Calvin cycle is where sugar gets made. In the cycle, carbon dioxide is fixed, which is this part of that chemical reaction that we talked about at the very beginning, and carbon dioxide is fixed to eventually be able to make glucose. There are three main stages to the Calvin cycle: Rubisco carbon fixation, reduction, and regeneration of RUBP. At the beginning of the Calvin cycle, carbon fixation occurs. A CO2 molecule combines with a 5-carbon acceptor molecule called ribulose 1,5-biphosphate, also known as RUBP, with the help of an enzyme called Rubisco, which is shown right here. And that results in this 6-carbon compound. But that's only a temporary state, as that 6-carbon compound is broken down into 2 PGAs, which are 3 carbon compounds. 3 carbon dioxides enter the Calvin cycle at once. So in actuality, as mentioned here, 3 of these 6 carbon compounds and eventually 6 of these 3 PGAs are made per cycle in the Calvin cycle. The next step is reducing these 3 PGAs into G3Ps right here, which are kind of like the step before glucoses. So these are 3 carbon molecules, just like the 3 PGAs, but they are reduced, meaning they gain electrons and through a series of reactions and eventually make G3Ps. And this reduction process takes up ATP. More particularly for every 3 carbon dioxides that go into the Calvin cycle, 6 ATP are used to reduce the 3 PGAs, and 6 NADPAs, so NADPHs, are also used to make the G3P molecules. This is why the light-dependent reactions that occur in the thylakoid membrane are so important. We are making the ATP and the NADPHs that are required for the carbon cycle. So after reduction, there's 6 G3P molecules, but only one of them goes to make glucose. 5 of the G3P molecules are recycled so that we can remake RUBP so that the Calvin cycle can occur and again and keep going. This process is called regeneration and also requires ATP. An interesting thing to note is that the number of carbons stays the same. So there's 5 3-carbon molecules, which is 15 carbons, and that is recycled eventually through the use of ATP and other chemical reactions into making 3 RUBPs, which are 5 carbon molecules, which again end up being 15 carbons. So the amount of carbon stays constant. And that is pretty much what the Calvin cycle is all about. It uses the ATP and NADPHs that we made during the light-dependent reactions in the thylakoid, and it uses those to fix carbon dioxides into G3Ps that go and make glucose. To make 1 glucose, the Calvin cycle actually needs to occur twice, because only 3 carbon dioxides go into it at once, and 1 usable G3P is released. So the Calvin cycle occurs once, 1 G3P is made, the Calvin cycle occurs 1 more time with 3 other carbon dioxides, and so you're left with 2 G3Ps, which are eventually made into glucose, which is a 6-carbon sugar. And all of this is occurring in the stroma, which is right outside of the thylakoid. So just as a quick summary, photosynthesis can be broken down into 2 steps: light-dependent reactions and light-independent reactions. In the light-dependent reactions, electrons are basically reduced over and over, and they go through the electron transport chain, such that at the end we make lots of NADPHs and lots of ATP. Those NADPHs and ATP are used up in the Calvin cycle to make G3Ps, which go and make glucose. And the main enzyme that helps with that is rubisco, as it fixes carbon dioxide through the Calvin cycle. Now let's talk about some complications. So the process that we just described with photosynthesis with the light-dependent and light-independent, that process applies to most plants, and those plants we call C3 plants, and they go through photosynthesis as described. So there is sometimes some complications that occur with the carbon cycle, where instead of fixing carbon dioxide, it fixes oxygen, which is a really big problem because the whole point of the Calvin cycle is to make these G3P molecules so they can go and make glucose. But if the Calvin cycle, if rubisco accidentally grabs onto oxygen, then the Calvin cycle burns up ATP and NADPH without really making something that's viable for the plant. That mistake is called photorespiration. So C4 plants, in order to avoid photorespirations, have 2 different distinct cells. They have the mesophyll cell that are located close to the stomata, where carbon dioxide and oxygen enter the plant. Remember, stomata are these little openings like on the underside of a leaf where gas exchange happens, and gases and like carbon dioxide and oxygen can go in and out of the plant, as well as water vapor as they are transpiring. C4 plants avoid photorespiration by using another enzyme called PEP during the step of carbon fixation, and this, again, as I mentioned before, takes place in the mesophyll cells that are located close to stomata. Another type of plant are called cam plants, and that's like the desert cacti, and here's my, again, weird rendition of cacti, and other plants that live in places with high heat, and in dry regions where they have to conserve water. They mostly absorb carbon dioxide at nighttime so that their stomata can remain closed during the daytime to avoid transpiration and loss of water. And that's what is an advantage of cam plants. And for C4 plants, their advantage is that they reduce photorespiration. Anyway, I hope you learned something new about photosynthesis, or maybe it makes more sense to you now. And yeah, thanks for watching.